The next thing that we're going to discuss in our genetics unit is DNA replication. There were a few questions yesterday when we were glossing over just the structure of DNA, and I'm glad that you were asking about how it's replicated. Okay, so each time the cell needs to divide in mitosis or meiosis, it needs to make a copy of its chromosome. Remember, a chromosome are just tightly wound pieces of DNA. It must be able to keep one copy while giving away. And by keeping a copy, it can keep making more and more copies of itself. And giving away some can further enhance cell division. This process of the cell dividing and DNA making a copy of its chromosomes is called semi-conservative replication. So in the end, you end up with one old strand and one new strand of DNA in every single cell. So in replication, basically, DNA is wrapped like the double helix, and what happens is helicase is an enzyme, and it unwinds DNA at a certain point by breaking those hydrogen bonds. Remember, hydrogen bonds are so sticky and they act like magnets. So they break the hydrogen bonds open between two complementary strands of DNA, like A and T. And then it unzips the helix. DNA polymerase is another enzyme that adds nucleotides to the free end, meaning it's going to make a copy of a free 3' end of the growing chain. Remember, adenine pairs with thymine and cytosine pairs with guanine. So DNA polymerase basically grabs onto that free 3' end and starts to build the new copy of DNA based on the template that's already there. Okay, some more about replication is that there's a leading strand, and there's in the leading strand, DNA polymerase goes from a 3' to 5' in that direction. The lagging strand will move on the other side of the DNA discontinuously, and it's making the other side of DNA. Because DNA polymerase can only add on at a 3' end, the lagging strand is what kind of goes almost like a broken train track and helps fill in the blanks of a broken train track. Then RNA primase attaches to DNA and synthesizes many short RNA primers. These primers are to show where DNA should be finished off, so DNA polymerase 3 can add nucleotides to the 3' end of the RNA primer. Now DNA polymerase 1 will remove the primer and replace them with DNA. So basically what's happening here with leading and lagging. Leading is really easy. The DNA polymerase connects at a 3' and it goes like a very smooth train track. Now the lagging strand is where DNA polymerase will attach at a 3', but that side does not really go in that correct direction, and so all these other enzymes are going to attach as flags and markers that are going to help create that other side of DNA to be made. So RNA primase attaches the DNA and makes little short primers. DNA polymerase reads those primers and attaches the nucleotides, and then DNA polymerase 1 removes the primer and replaces them with actual DNA. And then DNA ligase is the last part, and it inserts phosphates into any gaps into the sugar phosphate backbone, meaning that once the nitrogenous bases are made along with the sugar, then the phosphate can be added to that open side, and that'll make a continuous DNA strand.
DNA replication begins when helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the two complementary strands of DNA. DNA polymerase can only add new nucleotides to a free 3' end of a growing chain. Synthesis of one strand of the DNA, called the leading strand, proceeds continuously in the 5' to 3' direction. Synthesis of the complementary strand, called the lagging strand, is more complex. DNA polymerase can add new deoxyribonucleotides only to a free 3' OH. To provide a free 3' OH starting point on the lagging strand, RNA primase attaches to the DNA and synthesizes a short RNA primer. DNA polymerase III then adds deoxyribonucleotides to the 3' end of the RNA primer. DNA polymerase I replaces DNA polymerase III, removes the RNA, and replaces it with DNA. Finally, the enzyme DNA ligase forms a phosphodiester bond between the 3' OH of the growing strand and the 5' phosphate in front of it. During DNA replication, the leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously.
Okay, be sure to ask me questions about this tomorrow so that we can really get a clear idea about how the replication happens. So this is just a bigger picture of the replication fork. So here we have the leading strand, and like I said, like a train on a track, from the 3' to the 5' direction is where it attaches, and then we've got a train track that is worked really easily. Now on this side, it's going to be opposite, so it's not going to be a 3 to 5', it's going to be 5 to 3', because it is a complementary version of this side of the double helix. So what happens is the nucleotides are made discontinuously with RNA primase attaching at a 3', then creating RNA is a disposable copy of DNA, so it's like a little disposable copy of what should be there. That's further recognized by DNA polymerase, and then so real DNA is made from that recognition, and then DNA ligase comes and fills in all the blanks. So it's filling in phosphates here where there are different breaks. The lagging strand is going to make Okazaki fragments because that's the only way that the DNA can be created. On this side, it's going to be very continuously. On this side, it's discontinuously, and those discontinuous parts are called Okazaki fragments.